So what I do in neuroscience, and also, um, so basically my lab main focus is to build this big, incredible microscope in order to look directly into the mouse brain or other animals' brain. I do mouse stuff. That's why I say mouse. But you can see into a monkey's brain, a fish brain, C. elegans brain, like worm's brain. And the idea is like to see the neuronal activity and figure out what's going on in the brain and understand the algorithms that the brain is using in order to encode information and also decode the information like from its own encoding and then to perceive and be able to move, etc. So it's very interesting. And today what I'm gonna give you a sort of like a broad tour of how my things look like. I'm gonna explain to you like a small experiments that I'm running right now, and hopefully we get to the point that I can show you how to put the animals and we do the experiments. So I'm gonna share the other screen that I have. So what I do is just I look at the mouse brain directly, but the important thing is like to look at the mouse brain while the animal is executing a behavior. So I have a pretty parameter, like a pretty specific behavior because I'm studying how the brain encodes for time, for like for how the time is passing. So normally we are pretty aware of how much time has passed between two very important salient stimuli. And the idea is to try to understand how is the brain keeping track of this timing. So um, I train the animals to perceive two different time intervals. One is going to be short, so it's of half a second, and the other one is a long time interval of 2 seconds. For the animal, this is long. No, for us, it's very fast, but if you do it too long, then the mouse kind of get distracted. And so what I do in order to study that, now I'm going to share the screen of this thing here. Okay, yeah, you can see this screen. So this is the first time that I do it like this. So let me know if you have any problem. So what I'm going to use in order for the animal to basically um, perceive these two intervals of time and also make sure the animal is actually paying attention to this is I'm going to implement a system is called this is not working, 2-4 choice task. So the animal has to basically what is gonna happen is that I'm gonna give the animal these two different the stimuli, which is the interval, and it's given by applying one tone on a specific frequency at the beginning of one trial. And then the time interval is gonna pass between the when I apply the second tone. This is the second stimulus. So in the case of a short interval, it's gonna be half a second, as I say. And if it's a long interval, the second tone is gonna come later, no? At the 2 seconds around, okay? So the animal has to basically perceive these two tones, and then the animal has to tell me which tone, basically if it perceives a long or a short interval. So I'm gonna just present the animal to a water spout, so the animal can then, so if I have the mousey, like I have a mouse like here, the mouse is gonna perceive these two tones, and then it's gonna have two spouts where water is gonna come out of, and the animal has to, if it perceives by example, a short tone, the animal has to leak from one spout, and then it's gonna get the water reward. If it leaks from the other spout, then it's gonna get a punishment. It's gonna get a punishment. So, and if the animal perceives a long tone, then the animal needs to start leaking to the other spout. So that's why it's called a two-force choice task, because the animal has only two choices to make. So you're forcing the animal to make two choices, and it's either lick right or left. And in this case, by example, if it licks to the right, it's gonna represent that the animal perceive a short interval. And if it licks to the left, it's a long interval. So I'm gonna be training these animals these animals to be doing this. So if I go back to the Zoom and I start my camera here, okay, how can we move the camera? Okay, to the Zoom camera, to the iPad, yeah. So, okay, so here is it's basically the room where I work, and this is how basically the setup looks like. So I'm gonna push it out of this box a little bit. So you can see here, like the kind of things that I use. So this is sort of like a setup built out of optomechanical parts. These are mechanical parts that you use for like different like optical studies, how to build microscope, you use these kind of parts. And then I also custom made different parts like these. This is a spout holder, like this is like a valve holder. All of these things, what we do is we use 3D printing machines. So I design them, what I need, and then I 3D print them here in the university. And I also use some electronics. So in order to sense, which spout, these are the spout. Wait, I'm sorry. The lights turn off because I need to be normally in the dark. So whenever if the animal then perceive, for example, the short interval and it has to lick this spout, I can touch it and you will see the light down there of that capacitive sensor. That's a chip that is sensing the changes in capacitive. So it's gonna sense the touch of the spout or the touch of this other spout. And whenever, then I have like an entire like electronic board, like this is an Arduino board, maybe you guys are familiar with that, in which then it's gonna sense that one of the spouts is being touched. And if the right spout is being touched, I'm gonna activate these very tiny water valves. So they're gonna just deliver a drop of water. And everything is basically controlled by a computer. And I can see the animals using a regular webcam. And this is basically another valve that is going to deliver some air, because if the animal makes a mistake, I just punish them. So normally I have another setup that I'm building here. So this is sort of like the same idea, a little bit cleaner. So normally the animals will go inside this tube and I will head fix it here. And then the animal is gonna be exposed to the different stimuli, and then it can touch either one or the other spout in order to lick and get a reward using, and everything is controlled using these things, no? The Arduino board. So if you don't have any more questions, then I can also explain to you the rest of the experiments. Let me stop sharing this one.
Francisca, can I ask a question for a second? So
yeah.
You do need to have an engineering background, right, in order to do what you're doing, because I see you're designing some of the things, right?
Actually, I don't have an engineer background.
You don't have, okay.
I started biochemistry, and I honestly have no idea what I'm doing most of the time, if I'm completely honest.
Okay.
And I think that's the beauty of doing like a PhD or keep doing science, is that you're always learning new things. So my background is in biochemistry and molecular genetics. And then I started going into neuroscience, and I learned I learned about the brain. I learned how to build this stuff. And then it's like, or like how to execute behavior, and then you design this sort of paradigm and you have to build it. And once you are exposed to this challenge, you're like, okay, how do I tackle this? And then you ask around, and they told me, no, you use these kind of microcontrollers in order to control all this behavior. Okay, then I learn how to do it, I practice, and everything requires just learning new things constantly. So I don't have any engineer background. I just try to do my best. And the last time that I built like a solder any circuit was when I was in high school, and that was like, I don't know, 15 years ago or more. And I had to sort of like relearn everything and then ask around about different strategies that you can use, basically.
Okay.
So no, you don't need an engineer background. You just need to be, you just need to try to do your best, I guess.
Okay.
I think that's my point. So so then one thing that I want to do, so the animal, I want to show you one thing. So, okay, the animal is executing a behavior, and I also want to know whether there are specific parts of the brain which are important for this behavior. And does anybody know any technique that could tell you which brain area could be involved in, I don't know, in like executing one behavior, leaking, or like in my case, is keeping track of time? Anybody comes like thinks of like experiments that you can maybe do in order to figure out which brain area is involved? No, in the brain we have like different cortical areas, somatosensory, you know, you have motor cortex. Yeah.
Okay, Krishana, you wanna yeah.
Yeah, someone put it. Yeah, MRI was what I was going for. It's
You
like
can
you
do,
can like do activities based on like keeping track of time, and then
yeah,
scan the brain accordingly to see where it's, you know, where you can see activity in the brain.
exactly. There is a lot of people, there are some studies that they've done that with humans. So they put them in these fMRI scans and they look at which brain areas kind of like activate when you have a task that involves keeping track of time. And that actually something I read before like building my project and my hypothesis, and you come up with different brain areas that might be interesting to study. So I don't have an fMRI machine in the lab for my mice. That's kind of expensive. So, and like sometimes we don't need it. So how do you think I can, in a mouse, um, prove that whatever I'm looking in a human fMRI, it's translatable to the mouse brain? Any guess?
Oliver
Electrode
said,
patches?
yeah.
It can be electrode patches, but the problem is that the brain is kind of big, and I cannot just put electrodes everywhere. I have to select an area. So I'm not doing electrophysiology. I'm actually doing imaging, and I just put the microscope and look at specific areas, and it's basically the is like an electrode patches. I'm gonna record the activity of these neurons. But how do I make sure this is sort of like whatever I'm seeing in a recording that looks like these neurons are keeping track of time are actually important, and you need them to keep track of time? And about the mice, you normally buy them on companies. You know, these are genetic, very specific brand of mice. But how do you, if I see, by example, that in my case, I'm interested in an area called the striatum. So if I see that the activity here has some patterns that involve timing, how do I make sure that this is it necessary to have this pattern for the animal to execute the behavior? Any guess? Okay, so I'm gonna show you how ways that we do it. So one cool thing that we have available now is that if we have a neuron, so I'm gonna draw like a classic neuron.
Someone said open the mouse skull.
I do open the mouse skull, but then you see the activity. It's like, how do you make sure that it is necessary? Maybe you can deplete the neuron, the area, you can open the skull, you can remove the entire brain area that you're interested in and see whether the mouse can execute the behavior. But it's a little rough, no? Right now we have better technologies. So we have a neuron. It's kind of ugly. I'm so sorry about that. And neurons, they communicate and they fire these action potentials, no? You have like two action potentials coming out, no? And how do I know that this neuron is important for my behavior? What I do is I use a bioengineering tool. So I'm gonna express inside this neuron a specific receptor. A receptor that is not normally expressed in neurons, but I engineer. And these are receptors based on opsins, like the ones that we have in our eyes, okay? So opsin, basically, these receptors, the idea of this receptor, if I shine light, like a photon, to these opsins, then the opsin is gonna get activated. And what's gonna happen is it's gonna induce, you can either induce action potentials by opening these opsins and allowing sodium, again the light, sodium to come into the brain. And this is how you depolarize a neuron. Or in my case, what I'm gonna do is allow chlorine. And chlorine, what it does to the neuron is gonna hyperpolarization. It's gonna induce hyperpolarization. So what happened with that is that you are gonna basically eliminate the ability of the neuron to send action potentials. So it's gonna be flat, nothing is gonna happen. So the practical idea is that to a mousey, I'm gonna have a mouse. No, this is a very ugly mouse. I'm so sorry about that. Okay, to a mouse, I'm gonna, I have the brain of the mouse, and I'm gonna express the opsin in the specific area that I wanna do. And then what I'm going to do is when the animal is executing the behavior, I'm going to shine light directly here, the photons light, and I'm gonna inhibit that area of the brain. And so I'm gonna do that especially when the animal is like perceiving the time. And if I see a disruption on the behavior, then you can sort of like pinpoint the area and say like this area is actually necessary for the tracking of time. Because if I inhibit it using these opsins, I'm gonna disrupt the behavior, the ability of the animal to keep track of time. So this is actually a technique that was developed by bioengineers. So you basically take a protein that it's not supposed to be expressed in the brain, but if you put it in the brain, you can modulate the activity of the neurons. So you can either inhibit these neurons or you can activate these neurons. And that's sort of like a more precise way to modulate neuronal activity comparing to just, by example, removing some area or like applying some broad anesthetic in that area. So here I can inhibit the neuron specifically during the time the animal is perceiving the brain, the time interval. So if I show you here, do I see this? Yeah. So okay, so okay, so if I show you here, I have so like these are the optical fibers that I use to deliver the light in the brain. So here is sort of like what it drives my LED. There's a LED drivers, and what I'm going to show you is that whenever I want to basically inhibit the neurons, I have my fibers here. Okay, so this is hard because I cannot, I don't know where it is. Here, okay. And I can turn on these, and there's blue light coming out of this. So you can see blue light, and this light is gonna go directly into the mouse brain, and it's gonna inhibit the area specifically. So I'm using tools that are like meant for electronics and computers. So I have this blue light that I can again trigger with a microcontroller and then illuminate the brain directly and judge whether a specific area it's involved in the neural activity in perceiving time or it's not.
Francisco, we have like 2 minutes left.
Yeah, we have 2 minutes. So it's very short. I would love to show you how the animal does it, but the idea is that then you have the behavior and you can inhibit the brain specific area and determine whether you can sort of like, you need this area in order to execute the behavior. And the idea here is that for this neuroscience research, you use engineering tools. I actually use a lot of programming. So I program in Python a lot. All of this is driven in Python. And we use all these molecular tools in order to express these different opsins inside the mouse brain. And we also need to know a little bit of biology to know how the mouse behaves and how to treat animals and do surgeries and all this stuff. So this is very fast. I wanted to show you how you can actually inhibit the neurons, but you can imagine that the mouse goes into the setup, then the animal just perceive both. And if I turn on the lights, then the animal is not gonna be able to basically judge the time interval that it's being presented.